Okay, thank you very much for getting me an opportunity to present our work. For slide change, today we'll talk about the ways to visualize the protein families. Sorry, tech. Okay, so what actually do we expect from a good visualization tool of an ensemble of protein sequences? Is first that must be easily readable. Secondly, we need to easily identify potential clusters as with other projection methods. We would like to preserve as much as we can local similarities between the proteins, as well as ideally the global structure of the family. And it would be great that the tool would be rather fast and would not require our self-computational time. So for today, most of the time when we deal with protein families, that is the protein and its homologous sequences, found in one way or another, we either analyze the multiple sequence alignment directly, but it provides you information on the very local properties on the conservation of different positions, but it's very hard to deduce if there is any relation or any groups present in such data. Or if you want to go further, we can perform a phylogenetic analysis, and then we will have a complete idea of the evolution of the whole family and also establish the hierarchy present in the data. However, the phylogenetic tree constructions remain quite time-consuming, even for several hundreds of proteins. It can take several hours to days of computation, depending on the model that you put below. So the idea of our project was to develop a projection technique that will take as an input a multiple sequence alignment and provide an output to the interpretable projection of protein families. And we've been inspired by the work that was done by my friend and colleague Anna Klimowska, which is a projection methods developed initially for the single cell data. So for those of you who are not familiar with this work, I highly recommend taking a look. To develop a method named the Poincaré maps, which allows to represent the single cell data in the way that we clearly distinguish different clusters. We can also do some inferences about the cells in it. And here you can see that all the non-differentiated cells, the gray zone, will be always located close to the center. And the more you go to the border of the Poincaré disk, the more you will get the cell differentiated, as it also correlates well with the age of the embryo as shown in the other picture. And so since the problematic is quite similar, finding the way to represent of evolutionarily divergent proteins, we have decided to adapt it for the protein data. So what's the idea behind the Poincaré maps? The idea is that we take some points encoded by the features, and we will try to approximate the geodesic distances, so the distances on the Kynan graph, for example, by the hyperbolic distances, so by the distances in the hyperbolic space. So why hyperbolic space, and what is it? So Poincaré hyperbolic space can be described by a Poincaré ball. It's a Riemannian manifold, so it's very smooth, so it's easy to manipulate. And it's a distant metric defined on it, which depends on the norm of the vector. That is, if you can see here, all the triangles have the same surface and all the distances are the same, but the points located close to the origin have a smaller distances among each other, whereas when you go to the border, you can imagine if you're looking on the sphere like this, will have large distances as far as you get from the ball center. And it's a quite nice space to work with, because it can be thought as a continuous version of trees. So it's a very well approximation for the hierarchical data. It already has a distance metric in it, so we don't have to approximate it as we do in Tisneux with the Euclidean metric upon the embedding. And since it's very smooth surface, we can also do optimization using adaptive methods for the Riemannian spaces. So the computation, it will converge pretty well. And so the algorithm basically, without going too much into the details, would take your data encoded by the features. It will calculate some local similarities and construct a graph, kind of nearest neighbors graph on it. And we'll try to find the 2D projection in the hyperbolic space by minimizing the Kalbeck-Leibler divergence between the two spaces so that the distributions would be pretty similar in the projection and the original space. So in case of a single cell, your points are your cells, your features are your genes. In case of the proteins, your points are proteins, and we use the amino acid profile to encode for the protein features calculated using the Blossom matrix. And we use the cosine similarities as the initial distance metric to compare between the proteins. And that's what we get. So finally, the projection we obtain have a lot of nice advantages. You can see that clearly we can distinguish small groups. So here I present the results for the globin family. For those who don't know, globins are omnipresent in various organisms. And here on the left, I have colored them by their species. And on the right, by the phylogenetic clusters. That is, we have taken a phylogenetic tree and cut it in some threshold to see which proteins would be close from the evolutional point of view. And you can see that we clearly distinguish several groups which are associated either with the same organism or from the similar function. And what is even more interesting, for some of them, we would have different coloring in the projections. So you can see that on the left, you have some hemoglobins coming from bacteria and fungi, which on the right side are colored in the same color because they are evolutionarily related, and thus this kind of projection can provide us with information on the possible orthologs present in our data. So identification of the clusters is a nice property, but the main advantage of the Poincaré mapped projection is its capacity to reconstruct the hierarchy present in the protein data, and that we can see on the example of the thyroidexine proteins. So here is a study in which the authors have taken multiple sequence alignment of thyroidexine proteins, and using the phylogenetic inference have reconstructed several ancestral proteins. And they also have obtained some structures of it, so basically proven that these are the true sequences which probably could correspond to the last common ancestors. You can see there are some, the earliest stage will correspond to theoretical LUCA we don't have, but we have the last bacterial common ancestor, LBCA, last archaea common ancestor, LICA, etc., etc. And if you take the same sequence alignment as the author did and put it to the Poincaré MESA, it will produce the following projection. And if now we try to, as for globins, we clearly see some groups. Most of them correspond to some particular cases, so mitochondrial proteins or archaea proteins, which are well separated in clusters. But if we now take a look on the last common ancestors, so the reconstructed ancestral sequences, so most of them are located close to the center. And if you take a look on the distance from the disk center to each of the sequences, the order would be almost the same as we have for the phylogenetic tree. So basically, our method guesses what sequence to put in the root. It approximately corresponds to the root of the expected hierarchy, in our case, phylogenetic. Finally, this kind of projection can be extremely useful if we are talking about different annotations about missing or misannotated labels. And this I will show you on the example of the kinome. So kinome is an ensemble of kinase proteins, which are quite well studied and were annotated and re-annotated with the new knowledge coming. And here is an example of its projection in the latest annotation. And you can see that it's colored in the same way that the original science publication by Manning et al. And you can see that we basically reproduced the exact same topology of the repartition of different functional groups of kinases. It's just, you just need to swap symmetrically the disk to have green and yellow groups rearranged. And it's in a very good agreement. And moreover, if you now take a look on the previous annotations, our projection also makes sense. So if you take a look on the first, second, and third group, which are colored in gray, so gray in each projection corresponds to the unannotated sequences. So for 2 and 3, both groups were, with the succeeding studies of Manning and then of Dunbrack, were associated with the CIMK kinases. And they're clearly located close to this type of kinases in our projection. So that could be an indication even without this information. And in group number 1, there are some STO kinases which were wrongly attributed to this class in the UniProt annotation several years ago. And now were re-annotated in the correct way, which also, if you look at this projection, it became much clearer even without performing a much more complex and phylogenetic studies. And despite all the work done on the kinases, there are still several annotations which are missing in both annotations which we used. And actually, their proximity between each other makes sense. So there are several pretty distinct types of kinases, like a Hespin kinases, or those kinases located in the group 5, which, if you take a closer look, will correspond to the related genes, and so most probably make part of the same family. And also the group number 7 contains 2 different type of kinases, but which make part of the same monophyletic group. So potentially, well, in group number 4, there are 2 kinases which are not annotated but are clearly associated with the AGC type, which once again shows the potential of the clear visualization to help at least with the annotation of different proteins. So why not use other projection methods? Just a quick glance. So we have compared our results of Poincaré projection with Tisneux, MDS, UMAP, and PCE. And you can see that the data is not this complex. So basically, in all the methods, you will see some of the groups separated, but basically only Tisneux will give you any meaningful clusters. And which is even more important, none of this projection can inform you about the hierarchy of the data, that is the location of the ancestral sequence for tyrosine will be placed anywhere, and looking at any other projection, you cannot hypothesize on their role in the hierarchy. And if you try to quantify the quality of our embedding, Poincaré also performs better than others. Here we have used the scale-independent quality criteria. Basically, for each K, it will tell you at which point we reproduce correctly the K neighborhood. So if you reproduce only for small K, it's the case of Tisneux, we will very well preserve the local distances and local clusters. For K equal to the number of points, of course, we preserve everything. But you can see that for all numbers of K, the blue line dominates the others. It's Poincaré maps which generally preserve better the global structure. So you have two main parameters to play with if you use the Poincaré. So the first one is the nearest neighbors, and it works as it does in other methods. So the larger you took k, the huger will be the cluster. So more big and more redundant your data is, larger k you put. And there is also a parameter gamma, which serves for visualization purposes, which is the scaling factor in the Poincaré disk, which allows you to slightly sparse it from the border. And its main function is to help with visualization. You can also zoom. Well, I finished. Sorry. The zoom a given group to the center. So Poincaré disk is allows isometric rotation. That is without disturbing any distances. You can say, I want to take a look at the group here. So put it to the center without distorting any other distances. And all that you can do using the nice collab notebooks, which either take your MSA directly or the sequence that you love, which will pass by MMSec and will build the family for you and point you where your sequence is in the projection, or you can download the examples. So I will say just one word on the perspective, because we're still working on the project, and I hope that soon you will also be able to change the distance metrics and the protein representation to have to extract as much information as you can from your embeddings. And I thank a lot my collaborators and you for listening, and I encourage you to check the tool and the paper associated with it. Thank you.
Thank you so much. That was wonderful. Can we set up the next talk? And while we are setting up the next talk, does anyone have questions in the audience? Oh, yes. Sorry, I'll just...
Yeah, thank you. Very nice talk. You said that the map reflects the hierarchy in the data. Can you actually, is there a one-to-one relationship between a Poincaré map and a tree, a hierarchical tree?
I will just take the microphone. No, because Poincaré is a continuous version, and then the tree would be a discrete version, let's say, of the same type of hierarchy. And we cannot also, it's not always the case that it will be reconstructed correctly. It depends on how the data was prepared. If it's biased from one side, it tends to pull the center in the wrong way. So sometimes it's better to have any a priori and then rotate without in a symmetric way your map to put it in the center. Yes, and then, yeah.
Are you considering actually drawing a tree on top of the map? Would that be useful?
Yes, we're considering it's just technical. Yeah, it's a good idea, but we have not yet done it. We'll try to fit it on it. Yep.
Okay, yeah, thanks.